In this video, I will explain how a cytotoxic T cell identifies the infected cells and destroy them, kill them. However, please note that infected cells are not the only target cells for cytotoxic T cells. For example, cytotoxic T cells can identify and kill cancerous cells, transplanted cells, cells that receive from donors. But to be consistent throughout this video, I focus on how they identify the infected cells, differentiate these cells from healthy cells, and how they use some strategies to destroy only the target cell. Recall that inside the infected cells we have the microorganisms, and we refer to the antigen of microorganisms that are inside our cells as endogenous antigen. And also we recall that all our nucleated cells are capable of building a self-antigen named major histocompatibility complex 1. So as long as the cell is nucleated, it can build MHC-I, and we note that when cells are infected by microbe, they basically build a complex which is made of MHC-I and a piece of the endogenous antigen. Basically, this is a complex made of self-antigen and non-self-antigen. And as you see, the infected cells mark themselves. This is an official request from infected cells to cytotoxic T cells to come and identify and destroy them. So now it is not surprising that in the next image I can see an infected cell that is presenting its MHC-I antigen complex to the cytotoxic T cell. Cytotoxic T cell, using its antigen receptor, reads this MHC-I antigen complex. CD8 protein allows and helps the cytotoxic T cell to interact with the infected cell and read the MHC-I antigen complex. And to fully activate this cytotoxic T cell, we need a co-stimulator, which we know the major co-stimulator for activation of cytotoxic T cell is released from active helper T cells, and it's called interleukin-2. So now that cytotoxic T cell read and identified the MHC-I antigen complex and received interleukin-2 as the co-stimulator, the cell is going to multiply very rapidly. Proliferation of this activated cytotoxic T cell is going to happen simply because we have many infected cells in our infected tissues that cytotoxic T cells are supposed to destroy. So only one active cytotoxic T cell cannot destroy all those target cells. When cytotoxic T cells proliferate, we always get an army of two groups of cytotoxic T cells. One group would be active cytotoxic T cells or effector cytotoxic T cells. These are the cells that basically migrate to the infected tissues to identify the infected cells, and then by releasing different types of chemicals, they kill and destroy the infected cells. The other group that we get would be referred to as memory cytotoxic T cells. In this battle, memory cytotoxic T cells do not get involved, and that's the main reason they do not die. They will survive. So clearly that means when active cytotoxic T cells migrate to the infected tissues and identify and kill the infected cells, eventually these active cytotoxic T cells die. We do not keep them. After this battle against the microbe, the only cytotoxic T cells that we keep from this battle would be the memory ones. Now it's time to learn about a few techniques and strategies that active cytotoxic T cells use to kill the infected cell. For example, active cytotoxic T cells release a chemical named lymphotoxin. Lymphotoxin is going to stimulate some enzyme inside the infected cell. Those enzymes attack the DNA molecules that are located inside the nucleus of the cell. They make the DNA to break apart, and as a result, cell dies. So if you think about the nucleus of the cell as the brain of the cell, in a very simple way, we can say that it seems lymphotoxin causes this cell to become brain dead and die. Another chemical released from active cytotoxic T cell would be perforin. Perforin molecules form very short tubes and insert those tubes into the membrane of the infected cell. Through these tiny tubes, the extracellular fluid flows into the infected cell and cell bursts. Cytolysis happens. Another group of chemicals released from active cytotoxic T cells are Granzymes. These are protein digesting enzymes that when infected cells receive them, apoptosis in this cell is going to start. Recall, apoptosis is the programmed cell death. And it doesn't matter in what way the infected cell dies. Always please note that the pathogens that we have inside the cell will be free. So now in this infected tissue, we have lots of dead, dying infected cells, and also we have lots of free microbes. So it is important and crucial that we bring the vacuum cleaners, the phagocytes, to the area. And active cytotoxic T cells actually participate in this act by releasing a chemical named gamma-interferon. Gamma-interferon attract and activate phagocytes to the spot. So we can say gamma-interferon is a chemotactic factor. And the major phagocytes in human body are neutrophil and macrophage. So when neutrophils and macrophages get to this spot, they start phagocytosis of the remaining of these dead cells and also those microorganisms that are now free. Eventually, as I mentioned, the active cytotoxic T cells are going to die, and we are left with memory cytotoxic T cells, which in the future, when we are exposed to the same microbe, these cells use their expertise, because they have experienced meeting that microorganism in the past, to quickly identify, for example, the MHC-I antigen complexes, and very rapidly become active and proliferate. Their proliferation and building a new army of active cytotoxic T cells and new memory cytotoxic T cells is so fast that in many cases, this new army of active cytotoxic T cells are going to destroy those infected cells, those target cells, so rapidly that we do not even experience the signs and symptoms of this infection. And clearly, we lose these active cytotoxic T cells, and we are left again with the memory cytotoxic T cells. And as long as we keep memory cells, we can say that we have some sort of immunity against that type of microorganism.